Assalamu alaikum everybody. This is the second video on the chapter on coordination. In this, we are going to take it on from the first video, which was about neurons and all about the reflex action. Now, in this video, we're going to talk about the resting potential and the action potential and saltatory conduction. And if there is time enough, then we'll talk of the synapse. If we take giant squid axons and we can somehow measure the resting potential across the axon membrane. Now, the first thing that you have to do is an electron made from a glass tube pulled to a sharp tip and open at the end is filled with electrically conducting solution. Now, you can see this is the phospholipid bilayer. This is the inside of the axon, and this is the outside of the axon. Now, second, you have connected the wire to an amplifier. Third, the voltage difference between the electrode placed inside and the one outside is detected, and then this small potential difference is displayed on a computer screen. And what we find is that in an unstimulated neuron, that's why we call this resting potential, a potential difference of minus anything between minus 60 to minus 70 millivolts is detected. So I mean, we'll either refer to minus 60 or minus 70, but it differs from one organism to the other, slight difference. So here, as it says, minus 65 millivolts is observed between outside and inside. Now, this is the resting potential. But I mean, how is it maintained? This is what we need to understand. First, of course, you've understood what I'm trying to say is that the resting potential is the inside of the axon, which is at minus 70 millivolts as compared to the outside. But the main thing is this is, of course, the name, the resting potential. But how is this maintained? This is what we need to understand. First of all, we have to recap the sodium potassium pump. And this was, of course, the mnemonic for this was Nokia, and this is 3 sodium out. And this was 3 sodium out and 2 potassium in always. So this was what the mnemonic was. So 3 sodium, mostly the sodium ions are being pumped out and the potassium ions are being pumped in. Now you can see the pink, these are the sodium ones. So the sodium is being pumped out and this is the sodium potassium pump. So the sodium potassium pump is pumping out 3. So there are lots of sodium outside. Now, as you can see, this is the inside and this is the outside. So there's lots of sodium outside because sodium ions are being pumped out and potassium ions are being pumped in. So you can see the very nice, these are the potassium ions and you can see these are the sodium ions. So the sodium ions are more outside, but then we also have simple facilitated diffusion channels. And you see there are two types of channels. One is for the sodium, and you can see this one here. This is the sodium leak channel. And so the sodium, because it's more outside, it is diffusing back in. And this is, of course, facilitated diffusion. But you see there is only one sodium ion channel. But then what is happening to the potassium? The potassium ions are leaking out. But there are more potassium per unit area. If you look at in this diagram, there are 3 potassium channels, but only one for sodium. The sodium potassium pump, of course, are there, as we can see there are 2 of these. So we say the resting potential is maintained. The resting potential is maintained by 3 factors. Number 1, the sodium potassium pump. So the sodium potassium pump maintains that. Number 2, sodium ions are diffusing in. And number 3, potassium ions are diffusing out. But the most important point is that the axon membrane is more permeable to potassium. Now, how do we see that? we see that in this distance, you can see this distance, there are more potassium ion channels as compared to the sodium ion channels. So we say the axon membrane is more permeable to potassium. And that, of course, that as it is more permeable to potassium, it is going to have more potassium diffusing out. So the inside is minus 70 millivolts. Similarly, similarly here in another diagram, you can see the same phenomena, is that we have shown you the sodium potassium pump, which is this one. And then you can see how sodium is diffusing in, but then we show you the arrow for potassium is a much bigger arrow, so more potassium diffusing out. More potassium diffusing out. So the resting potential is maintained, number one, by the sodium potassium pump. Number two, by the fact that sodium ions are diffusing in, as you can see here. So these are special channel proteins for facilitated diffusion. But as more potassium is diffusing out, this creates a negative, and this is called an electrochemical gradient. And so the inside of the cell is minus 70 millivolts. This is how the resting potential is maintained. This is another diagram to show you how the resting potential is maintained, and you can see that there's a phospholipid bilayer, and this is the inside, and this recording microelectrode is just dipping into the just a little inside the cell, while this is the outside, and then you have this electrode which is just placed on the outside, and this one is the one which is just inside, and this measures the electrical difference across this membrane, and that is why this is inside is negative 70 millivolts. Now coming to the topic of action potential, it can be roughly divided into number one, resting potential, which I have colored in green. Number two, depolarization. And number three is repolarization. Now you can see here the voltage was minus 70 millivolts. Now this is going to go up to plus 40 millivolts, or even it can sometimes is plus 30 in some books. So basically it has to be 70 has to go to first 0, and then it has to go up to plus 30. Now this movement from minus 70 to plus 30 is called depolarization, and this is because sodium ions are going to move in, while after that plus 30 then the next stage has to take place, which is called repolarization, which is this part here, and that is because potassium ions exit. So the mnemonic for it is very easy. It's very easy to remember this. So again, a quick recap. Resting potential, and then here also resting potential. Then depolarization. And the depolarization is because the sodium ions are going to move in. There's special voltage gated channels which will open, and sodium ions will diffuse in. And then there is a max that it goes up to, and that's plus 30 millivolts. And then we start off with repolarization. And this is a small undershoot which is called hyperpolarization. So this repolarization is because potassium ions, the voltage gated potassium channels will open and potassium ions will diffuse out. So action potential is made up of, and this is only going to happen when there is going to be a stimulus here. At this point there has to be a stimulus. Something has to stimulate this neuron, and that is going to be resulting in this action potential, and the action potential is this area. And this is not going to be a very long, this is only going to be a few couple of milliseconds, 2 to 3 milliseconds. So this is the action potential, and then the resting potential is maintained. Here is another diagram to show you the details of action potential. Now, the basically another point which we have to talk about is the threshold value. There is a threshold value which is this point here, minus 65. So from a minus 90, it has to go across minus 65, only then an action potential is fired. But if there is a very small stimulus, then it might not fire an action potential and it just dies down and the resting potential is maintained. Another diagram showing you how there are failed initiations. There's a stimulus here, and then there is depolarization, and then repolarization, and then there is this refractory period, and again the resting state. And the time here you can see is only a very short while from here to here. And this is, please remember, in milliseconds. Now another diagram showing you the different stages. Now you can see here one is the resting state. All gated ion channels are closed. In depolarization, the sodium channels open. And as we said about the Nike, so Ni. And then in repolarization, sodium channels inactivated, potassium channels open. Hyperpolarization is that short while in which the sodium channels reset and close, potassium channels still open for a short while. That's the hyperpolarization, the point here. And then you can see here it's minus 55 is the threshold. Threshold must be crossed in order for an action potential to be fired. But what is important in this is the fact that this word refractory period means the time in which another action potential cannot be fired. It is refractory. The neuron is unable to fire another action potential. Now that is of two types. One is the absolute refractory period and the other is the relative refractory period. And we have to know it which part is the absolute one. This part is the absolute one. This is the part where there is the sodium in and the potassium out, the depolarization and the repolarization. And then this is the relative refractory period. Now the refractory period can be better understood if we can see what is the absolute and what is the relative refractory period. Now the absolute refractory period, after the neuron has generated an action potential, it cannot generate another one. Now sodium channels are inactive, will not open. Most potassium channels are open. This period is called the absolute refractory period. But in the relative refractory period, which is this portion, you can see immediately after the absolute refractory period, the cell can generate an action potential, but only if it is depolarized to a value more than the normal threshold. And this can only happen if there is some very strong stimulus. And that is why this is called the relative refractory period. Now coming to the topic of saltatory conduction, you can see this is a myelinated axon. A myelinated axon speed is 100 meters per second. Now, how is this possible? In a non-myelinated, it's much lesser. The reason being is that we say it jumps from node to node. jumps from node to node. Now you know the area which does not contain myelin sheath, these areas, where there is no myelin sheath, that is the node of Ranvier. So what we have to understand is that when there is a stimulus anywhere here, say there's a stimulus here, now there is going to be depolarization. So when there's going to be depolarization, the inside is going to become positive and the outside is going to become negative. Now you see a local circuit is established. And this is how the action potential is propagated and electrical impulse is fired. So we say the fact that we say it jumps from node to node, the reason is because you see when there is a stimulus here, there is going to be from minus 70 millivolts, it's going to go to plus 40 millivolts, plus 40 or plus 30. So when the inside becomes positive and the outside becomes negative, we have a local circuit established. And this local circuit results in the transmission of the action potential. So the action potential travels across this, and then it's going to only travel in one direction because this is where the recovery is going to start. So as it moves forward, the area behind it starts to recover and the action potential is propagated. Another diagram showing you the conduction in a myelinated axon. As you can see here, this is the yellow is the resting potential, the blue is the depolarization, and red is the repolarization. So as the action potential is traveling, then the area behind it is repolarizing or is recovering. So myelin prevents the movement of sodium and potassium through the membrane. Nodes of Ranvier contain voltage-gated sodium and potassium channels. Saltatory conduction leaps. That's a very fast rate of conduction. Another diagram showing you the saltatory conduction in myelinated axons. So it's a very brief recap. An action potential in a myelinated axon produces electrical gradients in the intracellular and extracellular fluids that are similar to those observed in unmyelinated axons. Because very little current flows across the membrane where myelin insulates it, the current must flow all the way to the next round of Ranvier, where it depolarizes this area of the membrane to threshold and initiates an action potential. The jumping of action potentials from node to node is the basis of the term saltatory conduction. In a non-myelinated axon, as you can see, the speed is only 0.5 meters per second, while it was 100 meters per second in a myelinated neuron. The reason is that whenever there is a stimulus here, this will become positive. The inside will become positive, and the outside will become negative. Now, the local circuit is established like this. So it's not going to be jumping from node to node, but it's going to be very slow movement. And this, of course, is going to be resulting in the speed being much lesser because there is no myelin sheath. And so it's going to be local circuits being established. And as it is moved forward, the area behind the action potential is going to recover and the impulse is going to travel forwards in a certain direction. Another important topic is the intensity of the stimulus. Like, so if you had a very severe pinprick or a slight pinprick, now the amplitude remains the same. By amplitude, I mean the fact that from the resting potential of minus 70 millivolts, it goes up to plus 30 or 40 millivolts. Now this is the amplitude. This is not going to change. But the frequency changes. Like we had 1, 2, 3 action potentials. Now we have 1, 2, 3, 4, 5 action potentials. So this would be a weak stimulus, and this would be a strong stimulus. So a weak stimulus, and this is called the all or nothing law. That is, either an action potential is going to be fired or there's going to be no action potential. So the amplitude will always remain the same. But as the intensity increases, if it's a stronger stimulus, then the frequency is going to increase. And the number of neurons excited is also going to be affected. So the number of neurons, and the brain is going to perceive this as, is it a weak stimulus or a strong stimulus? Because the number of neurons stimulated will also affect the perception of the brain. Now we come to the topic of synapses. And synapses are areas between a microscopic gap between two neurons. Now each synapse is made up of a presynaptic neuron. So the action potential is arriving in the presynaptic neuron. And then there is a gap which is called the synaptic cleft or the synaptic gap. And this is a microscopic gap. And this is this area which I have drawn. And after that is the postsynaptic neuron, just like we have pre-partition or post-independence of Pakistan. So pre is before and post is after. So presynaptic neuron, which is this one which I have made. And then we have the postsynaptic neuron, and in between is a gap. Now the presynaptic neuron contains special channel proteins for calcium. So these are the calcium channel proteins which are present in the presynaptic neuron. But there's no such thing on the postsynaptic neuron. On the postsynaptic neuron, which is this one, this is the postsynaptic neuron. So this will be the postsynaptic membrane. There are special channels which are sodium channels. And these sodium channels are usually closed. So these sodium channels will only allow sodium in when they open. So the sodium channels are found on the postsynaptic neuron, and the transmission of impulse is always in this direction. Why? Because the presynaptic neuron has the calcium channels, plus it has these vesicles, and these vesicles contain a neurotransmitter. These vesicles contain a neurotransmitter. That neurotransmitter, abbreviation we write ACH, but actually it is acetylcholine. So the name of this neurotransmitter is acetylcholine. Now this is the structure of a synapse, and as we go ahead we're going to study how this synapse is going to function. Now this is a very good diagram to show you the synaptic transmission. So this is the presynaptic neuron, and action potential arrives. Number 2, calcium channels open and calcium ions diffuse in. Calcium channels open and calcium ions diffuse in. Now this would be facilitated diffusion. Now when calcium ions diffuse in, these cause the vesicles to move. These vesicles which are present here, they're going to move, and they're going to fuse with the presynaptic membrane. And then there is going to be exocytosis. And the chemical, which is acetylcholine, is going to diffuse out across this cleft. So it's going to diffuse across the cleft, and then it is going to attach to the ligand gated channels. These ligand gated channels were closed, but when the acetylcholine binds to it, they open. And when they open, sodium ions diffuse in, and if enough sodium ions diffuse in and the threshold is crossed, that will result in an action potential in the postsynaptic neuron. This is another diagram to show you the synaptic transmission, and it's very well documented. Number one, transmitter is synthesized and then stored in vesicles, which you can see here very clearly. Number two, an action potential invades the presynaptic terminal. Number three, depolarization of the presynaptic terminal causes opening of the voltage-gated calcium channels. Number four, influx of calcium through the channels. Number five, calcium causes the vesicles to move and fuse with the presynaptic membrane. The calcium causes the vesicles to move and fuse with the presynaptic membrane. Number six, now transmitter is released into the cleft by exocytosis. This word is very important that you know this word, exocytosis. Number seven, transmitter binds to receptor molecules in the postsynaptic membrane. So that acetylcholine which has diffused across this cleft will going to bind to the receptor molecules in the postsynaptic membrane. Opening or closing of the postsynaptic channels, both can occur. And the postsynaptic current causes excitatory or inhibitory postsynaptic potentials that change the excitability of the postsynaptic cell. And then we have an enzyme inside this, which is called acetylcholinesterase. Acetylcholinesterase is an enzyme which is going to break up the acetylcholine, and that is going to diffuse back into the presynaptic neuron, and then it is going to reassembled and then be again reconstituted into a vesicle and be ready to be used again. So the enzyme acetylcholinesterase is very important in the synaptic cleft, which is going to break the acetylcholine into acetate and choline. So removal of the neurotransmitter or enzymatic degradation, then retrieval of vesicular membrane from the plasma membrane. So this is again going to be retrieved and is again going to be reconstituted the vesicles. That is what we need to revise this once more and go through this again and learn all the stages of the synaptic transmission. And that will be all for this video. Thank you. Thank you for watching this video, and please do leave a feedback of any comments or any suggestions which you would like to make.